Alright, last PowerPoint we ended looking at hair. Now let's look at nails. Nails are just scale-like modifications of the epidermis, so they also contain keratin. They contain hard keratin like that of hair, so it's a tougher keratin as far as the fibrous integrity. It acts as a protective cover for the distal dorsal surface of fingers and toes, so that material that makes up your toenails and your fingernails is for protection. It consists of areas called a free edge, which is the part you cut, a nail plate, which is the part that goes over the nail bed, and then a root, which is actually not visible to you if you're looking at your fingernail. The nail bed itself is the epidermis underneath the keratinized nail plate, and again, the nail plate sitting on top of that nail bed. The nail matrix is the thickened portion of the bed responsible for nail growth, and it's closer to that root. Looking at nails, we have even more terminology. There's nail folds, which are skin folds that overlap the border of a nail. There's the eponychium, which is a nail fold that projects onto the surface of the nail body. You probably would be more comfortable calling the eponychium the cuticle. Then you have the hyponychium, which is under the free edge or under the part you cut, so this would be the area where dirt accumulates. And then nails normally appear pink, but they can appear a little bit more opaque. And the reason that they would normally appear pink, however, is because of underlying capillaries or blood supply in that nail bed. In most nails, there is a lanule, sometimes called a lanula, and this is thickened nail matrix that appears white closer to the eponychium than the hyponychium. Now, an abnormal color or shape of lanule could be an indicator of disease, but not necessarily. Also, some individuals are missing lanule, and that doesn't mean anything is wrong with them. So let's look at an image showing you these structures. Let's start with the top. So lanule or lanula is a crescent-shaped region close to the eponychium. The eponychium is going to be the cuticle. Then you have lateral nail folds or pieces of skin that fold over the nail plate, which is usually pink in appearance, and you have the free edge of the nail, which is what you cut. Under the free edge of the nail is the hyponychium, which is the region where dirt can accumulate. Here's just another view of the nail plate, under which is the nail bed. So that's where there is some vascularization or blood supply that gives that nail bed and that nail plate a pink appearance. We can even see the phalanx or the bone of the fingertip. And then you have, again, another view of the cuticle of the eponychium. We have the proximal nail fold, which is going to be above the nail root, so that's a thickened region of the nail itself, and then around the root of the nail is the nail matrix, which is thickened connective tissue, well, epithelial tissue surrounded by connective tissue, from which you have mitotic cells that produce more keratinocytes to create the nail. But we can use nail appearance to diagnose some diseases, so even though these different appearances maybe something you have, it doesn't mean you have a disorder, but you might want to consider getting it checked out. If you have yellow tinged nails, this could indicate a respiratory or a thyroid gland disorder. If you have thickened yellow nails, this could be due to a fungal infection. If you have a spoon nail, so kind of an outward concavity of the nail that could signal an iron deficiency, so some type of anemia. This spoon nail is called chalakachia. Then if you have Bowes lines, which are horizontal lines that cross the nail, this could indicate illnesses such as uncontrolled diabetes or a heart attack or even the result of cancer chemotherapy. Here is that spoon nail, so you have kind of that central concavity, the chalakachia. Maybe pronouncing that wrong, that's just always how I pronounced it. Next we're going to be looking at sweat glands. Another name for sweat glands is sudoriferous glands. All of your skin surface, except for a couple locations like your nipples and parts of the external genitalia, are going to contain these sudoriferous glands. So you have about 3 million per person. There's two main types. There's eccrine, which are just merocrine sweat glands, and there's apocrine sweat glands. Now we find inside the eccrine and apocrine myoepithelial cells, and these are not muscle cells, but they are epithelial cells that have some type of contraction function. They contract upon nervous system stimulation, and they force the sweat into ducts. So there's some contractility to these myoepithelial cells, but they are not muscle cells. We're going to look at eccrine sweat glands first. Again, these are just merocrine sweat glands, and they're the most numerous type. So you find them in thick skin, on the palms, soles, and also in your forehead. Your forehead doesn't have thick skin, but palms and soles definitely. These types of sweat glands have ducts that connect to pores, and they function mainly in thermoregulation. So they're going to be controlled by the sympathetic nervous system. Their secretion you would call sweat, and it's mostly water. Most sweat is water. There's some salts, other ions, things inside sweat glands like vitamin C, antibodies, dermacidin, which is antimicrobial, and some metabolic waste, usually nitrogenous. So again, we're talking about eccrine sweat glands. Eccrine sweat glands are able to secrete their product from the gland itself into a duct, and the duct goes to a pore. The pore could be on the surface of the skin, or if we're looking at a sweat gland in the human body, it could go to inside an organ, but it's to the surface of an epithelial layer. Apocrine sweat glands don't quite function that way. Apocrine sweat glands are confined to auxiliary or anogenital regions, so apocrine starts with A, and the locations of the sweat glands also start with A. So it's confined to areas like your armpits and around your genital areas. Their type of sweat is still going to be made up of some water, but it's a little more milky, it's a little more thick compared to eccrine sweat. This sweat that comes from apocrine sweat glands is not only milky, but can be yellowish sweat, and it's because it contains fatty substances and proteins. And bacteria are able to break down this milky yellowish sweat, and that's what leads to body odor. Apocrine sweat glands are much, much bigger than the eccrine sweat glands. And their ducts don't empty to a pore on the surface of an epithelial sheet. Instead, their ducts empty into hair follicles. They empty hair follicles. And then from those hair follicles, the secretion kind of seeps up around those hair follicles. The apocrine sweat glands do not begin functioning until puberty. We don't really know what their function is, but we think it has something to do with pheromones, basically acting as a sexual scent gland. There are some modified apocrine sweat glands, which really don't act like apocrine glands anymore, but there's ceruminous glands, which line the external ear canal, and mammary glands. The ceruminous glands secrete cerumen, which is just earwax, and mammary glands secrete milk. So we'll move away from the eccrine glands. So eccrine glands and apocrine glands, those are both sudoriferous glands, which are sweat glands. Sebaceous and oil glands are what we're going to look at next. These are also widely distributed, but we don't find them in the palms or the soles or where thick skin is. We would see that the sebaceous or oil glands produce sebum. They're going to produce this sebum and release it next to hair follicles, kind of like the apocrine glands. And they're also relatively inactive until puberty. They're usually stimulated by hormones, especially androgens like testosterone. And this oily holocrine secretion is going to contain dead cell debris. That's why it's a holocrine secretion. This isn't a merocrine gland. And there are some bactericidal or bacterial killing properties in the sebum, but its main function is to soften the hair and skin. So let's look at eccrine glands compared to a sebaceous gland, and then I'll also point out an apocrine gland. So here is a histograph of a sebaceous gland. It's alveolar in structure or sac-like, and it's going to secrete its secretion to the canal, usually called the biliary canal, which is next to a hair follicle. Here's another picture of the sebaceous gland. We also had apocrine sweat glands that secreted their secretions to the biliary canal next to a hair follicle. And that was a sweat gland as opposed to an oil gland, which is what a sebaceous gland is. And then eccrine glands weren't near hair follicles. They were in the dermis like the other glands, but they secreted their secretion up to a pore, so into a duct up to a pore, which went to the surface of an epithelial sheet. So we have some differences in secretion here. We have some differences in location. For example, let's look at eccrine, apocrine as sweat glands, and then sebaceous glands, which are oil glands. Both types of sweat glands are going to be merocrine, meaning that exocytosis is the main way in which the cells that make up these glands secrete their product. We're not going to see dead cell fragments or apoptosis of the cells when they secrete their product. We see in eccrine sweat glands a duct that goes to a pore, whereas we see in apocrine sweat glands we see a duct that goes to the biliary canal next to a hair follicle. And same location for sebaceous glands, for their duct and where they secrete, as there is for an apocrine sweat gland. But we don't have this coiled tubular structure for sebaceous glands. Instead, sebaceous glands are sac-like or alveolar. The secretions for sweat glands or sweat is a little bit thicker and yellowy and milky if we're talking about the sweat that comes from apocrine sweat glands, whereas oil comes from sebaceous glands. That oil is generally called sebum. And I'm just going to go through this one more time because it's important. Secretions exit the ducts of these glands at the skin surface for acrine sweat glands, usually in the upper part of the hair follicle or biliary canal, rarely, if ever, the skin surface for apocrine sweat glands, and then usually the upper part of the hair follicle, sometimes the skin surface for sebaceous glands, but usually again that biliary canal. And we can find acrine sweat glands pretty much everywhere. You can find apocrine sweat glands in the armpits around the genital region, and then you can find sebaceous glands almost everywhere. You can especially find acrine sweat glands on the palms and soles of the feet, on your forehead, and you can never find sebaceous glands on your palms or soles of the feet. So there are some differences into where we can locate these glands as well. A homeostatic imbalance associated with these glands is acne, which is just infectious inflammation of sebaceous glands, so your oil glands, that results in pimples, which are usually pustules or cysts. Usually these types of pustules or cysts are infected with Propionibacterium. That type of Propionibacterium can create an acne infection. Whiteheads are going to be blocked sebaceous glands, and then blackheads are whiteheads that have been oxidized. So whitehead can become a blackhead. If you have overactive sebaceous glands, meaning a high production of sebum in infants, that can lead to seborrhea, which is known as cradle cap. So it really begins as kind of a pink rash with raised lesions on the scalp, but those raised lesions can turn yellow-brown as they heal and flake off. So here's a newborn with cradle cap or seborrhea. Skin is going to be the foremost barrier, so it has a lot of functions that match the fact that it is a barrier. It is protective. It's going to be responsible for cutaneous sensations from external environments. Things you might not have thought about if you've just been thinking of it as a barrier, however, include a body temperature regulation, metabolic functions, acting as a blood reservoir, and excretion of waste. The protection function makes a lot of sense. It's made up of many layers of flat cells, if we're looking at its epidermis, and then if we count its dermis, there's a lot of tissue to get through before a pathogen or something that causes a laceration gets to deeper, more damaging tissues. Body temperature regulation, that has to do with moving blood in and out of dermal capillary loops. Cutaneous sensations, there's tactile discs, Meissner corpuscles, tons of different receptors that react to different neurons. Metabolic functions is going to help create vitamin D. Vitamin D is found in your skin, and that's going to be activated and move throughout the body to become calcitriol. So blood reservoir, at any one point in time, you might find up to 60% of the blood in the dermal capillary loops. And then excretion of waste. Some waste, like nitrogenous waste, can be evaporated in sweat out of the body via the skin. So let's look at protection first. The skin is exposed to microorganisms, abrasions, temperature extremes, harmful chemicals, sometimes on a daily basis. And therefore there are not just a physical barrier that we're alluding to for the protection provided by the skin, there's the chemical barrier and the biological barrier. The chemical barrier is talking about the chemicals secreted from glands, so sebum or oil, defenses, which is antimicrobial, sweat, which also has even more antimicrobial proteins, all of these chemicals that are secreted to the surface of the skin. This creates an acid mantle or a low pH of the skin, and not only do the antimicrobial proteins impede bacterial growth, the low pH of the skin retards bacterial multiplication. Melanin also provides a chemical barrier, and that chemical barrier is against UV radiation damage. The physical barrier is easy to understand. You have many layers of flat dead keratinocytes. So there's many layers of cells before you even start to damage living cells. And then some of the chemicals that could come or be put on your skin have very limited penetration of the skin. It's hard for lipid-soluble substances, other plant lipids or oils, organic solvents, salts, drugs, even drug agents to get too far past the water-soluble or water-insoluble lamellated granules. If we're talking about a biological barrier, the epidermis contains phagocytic cells, which are things like macrophages. They're going to be able to eat up debris and pathogens. Dendritic cells are also in that basal layer of the epidermis. They're very good phagocytic cells. They're also good at alerting the immune system. So the dermis contains plenty of cells that are able to act against pathogens or act against cell debris and help alleviate injury or inflammation. Also, DNA can absorb harmful UV radiation, and that can convert it to harmless heat. However, we don't want our DNA to do that too much because that can cause it to mutate and cause skin cancer. A homeostatic imbalance we want to talk about quickly is just if organic solvents or heavy metals pass through the skin and enter the blood. There are some chemicals that when mixed with things like DMSO can then travel straight through the skin. And these things can be very fatal to multiple organ systems. They can be very devastating. Things like the kidneys can shut down, the brain can be damaged. Absorption of lead, for example, can result in anemia and neurological defects. So we gotta be careful when we're using anything that has an organic solvent in it or a heavy metal that can very easily be dissolved all the way through the skin. So let's talk about some of the other functions besides protection. There's body temperature regulation. So under normal resting body temperature, sweat glands produce about 500 milliliters a day of unnoticeable sweat. And if sweat is unnoticeable, even though it's 500 mils per day, we're calling that insensible perspiration. Now, if body temperature rises, so think Georgia summer, you're going to see dilation of dermal vessels, and that's going to bring heat to the surface of the skin, and that can increase sweat gland activity. So you can start to produce upwards of 12 liters or 3 gallons of noticeable sweat. And if it's noticeable sweat, we're going to call that sensible perspiration. And sensible perspiration is designed to cool the body through the property of water called high heat of vaporization. If you need to remember what high heat of vaporization is, go back to chapter 2. There's also the fact that sometimes the environment's not warm. Sometimes the environment's cold, and a cold external environment wouldn't cause the dermal capillaries to dilate. It would cause them to constrict, and that would send blood to deeper tissues, closer to your vital organs. But that would mean your skin temperature drops, and that would slow passive heat loss. Cutaneous sensation, I talked about Meissner's corpuscles, just any type of exterior receptor that's responding to stimuli outside the body. Usually it's temperature or touch. Thermoreceptors are the temperature receptors, but there's various different types of touch receptors like the Pacinian corpuscle, Meissner corpuscle, tactile disc. There's even free nerve endings that sense painful stimuli, and they can be found mainly in the dermis. So looking at the skin structure, we've looked at hair follicles, we've looked at glands, also found in the dermis. We've talked briefly about temperature regulation via blood vessels. And now we're talking about the nervous structures that sense tactile sensation. Things like free nerve endings or sensory nerve fibers like the lamellar corpuscle. Those are going to be able to pick up on things like touch. So metabolic functions, mainly synthesis of vitamin D. You need vitamin D for calcium absorption in your small intestine. There's different chemicals in keratinocytes that can disarm some carcinogens. That's not going to necessarily be able to stop all cancer-causing agents, which is what a carcinogen is. Instead, keratinocytes are a little bit better at activating some hormones like converting cortisone into hydrocortisone, but they also make collagenase, which aids in natural turnover of collagen to prevent wrinkles. But the biggest metabolic function I want you to pay attention to is the synthesis of vitamin D. Now, it's not completely activated in the skin, but it'll go through a series of steps to become calcitriol. It's also a blood reservoir. The skin, especially its venous structures, can hold up to 5% of the body's total volume. If you think about venous structures in general, they can hold up to 60% of the blood's volume at any one point in time. So the skin is just a small part of that blood reservoir. So So skin vessels can constrict or dilate in order to hold more or less blood. So they may dilate in order to hold more blood or constrict in order to shunt blood to other organs, and that would occur during exercising. Excretion, the skin can excrete nitrogenous waste. Usually those nitrogenous wastes are things like ammonia, urea, uric acid, but sweating can also cause salt and water loss, which that can be dangerous. Our next topic is going to be skin cancer and burns. So we're going to be looking at characteristics of these two ailments that ail the integument. And the skin can develop over 1,000 different conditions and ailments, but these tend to be a little less common, but the most damaging. So even though there's many internal diseases that reveal themselves on the skin, the most common disorders and infections would take an unknown amount of time to go over. So even though we're looking at these less common types of illnesses, they are very damaging, and you should know kind of the clues for when to seek medical help. So we'll start with skin cancer. So cancer in general is a group of diseases, not just one. We have different skin tumors, some are benign, not cancerous, and others are cancerous and spread. So cancers or tumors that spread are called metastasizing tumors. And risk factors for developing metastasizing tumors include overexposure to UV radiation or frequent irritation of the skin. So even some skin lotions can contain enzymes that repair damaged DNA. Now, these are kind of few and far between, and you're not necessarily going to be using skin lotions to treat skin cancer. So there's three major types of skin cancer: basal cell carcinoma, squamous cell carcinoma, and melanoma. Basal cell carcinoma, you could say, is the least dangerous, not that it's not dangerous at all, but basal cell carcinoma is the least malignant, which means cancerous, and it's the most common. But being the least malignant, it doesn't like to spread to other areas. It's the stratum basal cells that proliferate and slowly invade the dermis and the hypodermis, so it's the stratum basal cells that are affected. You can cure basal cell carcinoma just by surgical excision in 99% of cases, and that's why it's the least dangerous, because excision itself usually can take care of it. So here's a basal cell carcinoma tumor on the left side of this individual's nose. Then there is squamous cell carcinoma, which is the second most common type, and it can metastasize, so it's a little bit more dangerous. It involves the keratinocytes of stratum spinosum as opposed to the squamous cell carcinoma, the basal cell carcinoma we just looked at, which involves stratum basal. Usually in squamous cell carcinoma, you're going to have also a reddened papule, but it's more likely to be located on the scalp, ears, lower lip, or hands as opposed to, for example, the side of the nose. But as long as you catch squamous cell carcinoma early, pretty good prognosis that it hasn't spread to other areas and can be treated not only by surgical excision but also by radiation therapy. So squamous cell carcinoma, a little more red, a little more pustule looking, a little more scaly, really common on the scalp. And then melanoma is the most dangerous type of skin cancer. Melanoma is cancer of the melanocytes, so its name means basically melanocyte tumor. It's the most dangerous type of skin cancer because it's highly metastatic and it's resistant to chemotherapy. And so it would need to be treated not only with immunotherapy or radiation therapy or even different types of targeted chemotherapy, but with a really wide surgical excision to make sure all the cancerous cells have been taken out. The key to survival for early detection for melanoma, so make sure you know the ABCD rule. So if something happens to a pigmented area such that it changes its shape and suddenly is asymmetrical, or the two sides of a mole or a freckle suddenly don't match where they used to, or if there's a border irregularity, if it suddenly has an indentation where it previously didn't have an indentation, that would be something that you want to get checked out for possible melanoma. If there's a color change, this is a big one. If there's a color change, especially if it contains several different colors like black, brown, tan, or even like a red or blue, that's a big indication it might be melanoma. And then if the diameter is larger than 6 millimeters, not just in size of what's viewable, but like deep as well, it starts to feel like a nodule. So if it's larger than the size of a pencil eraser, something you want to get checked out, especially if it grew to that size, wasn't initially that size. So here is a photograph of melanoma. This is in the lower left ear. This is quite a advanced stage melanoma. However, if it hasn't metastasized, it's a pretty good prognosis that excision around this tumor could get rid of all the cancer cells. Next, we're going to be looking at burns, and burns can be caused by a lot of things: heat, electricity, radiation, certain chemicals. But that damage can cause denaturation of proteins, and that destroys cells. And as these cells are destroyed, they lose a lot of their fluids, and that leads to a threat of immediate dehydration and electrolyte imbalance. So the immediate threat caused by burns is not the damage to tissues, but it's the dehydration and electrolyte imbalance that can lead to renal shutdown and circulatory shock. So there's ways that we can evaluate burns. So we use the rule of nines. The body is essentially broken into 11 sections. Each section represents 9% of the body. You have to account for front and back, and then the genitals is going to account for 1%. And this is just used to estimate the volume of fluid loss. Let's talk about the different types of burns. You have first degree and second degree. First degree and second degree have really similar symptoms. They both usually have localized redness, some swelling, some pain, but you never have blisters with a first degree burn, whereas you would have blisters with a second degree burn. Also, with a first degree burn, you only have the epidermis damage. With a second degree burn, you have the epidermis damage and part of the dermis damage, usually the upper part. So a second degree burn is sometimes called a partial thickness burn. With a third degree burn, you have not only the epidermis, but also the entire thickness of the dermis, or you could just say the entire thickness of the skin is involved in a third degree burn. There may be a couple of blisters in areas where, or on the outside of the burn, where it's a little bit more of a second degree burn, but third degree burns are not characterized by blisters. Instead, they're characterized by no initial pain because the nerve endings have been destroyed, no edema because there's so much tissue damage, and the skin color is generally going to be gray, white, cherry red, or blackened. So here we can see, for example, a first degree burn on the fingertips, just kind of red, maybe slightly swollen. Down here's a second degree burn, so we have blisters, but still red, swollen. Third degree burn, we have that cherry red, and there would be no initial pain again because the nerve endings have been destroyed. This is something important to note. Burns are considered critical if over 25% of the body has second degree burns. Burns are considered critical if over 10% of the body has third degree burns. And burns are considered critical if the face, hands, or feet bear third degree burns. Treatments for burns are debridement, so you have to remove the burned skin, treat with antibiotics to prevent infection. Some burns, like especially third degree burns, may require a temporary covering. You may have seen some weird articles about them using tilapia skin as a temporary covering. It actually works pretty well. And even skin grafts may need to be required if it's a very damaging burn, so more common in third degree burns. Here is the figure showing you the estimating amounts for the rule of nines. So you can see just general areas. We have 4.5 on the front of the arm, 4.5 on the back. So front and back would be 9 for the upper limbs. Front and back of the head would be 9. Then front of the legs, so whole lower limbs, would be 9 and 9 on the back. And if you added everything together, you'd get 100%. This is where we're getting the percentages for when burns are considered critical. And that is it for Chapter 5, so make sure you watch the first and second half of the video lectures.